Hello friends, welcome back in the channel of Clinical Care Insight. We started a series of lectures on neuroanatomy and neurophysiology. Okay, until now we discussed so many topics. Okay, and before the break we are discussing about the synapse. We started discussion over the synapse, and we were seeing that synapse means a junction. Synapse means a junction between the two neurons. Okay, and if we talked about the outside the central nervous system, that it includes some of the autonomic, you can say, the connections, as well as the NMJ, neuromuscular junctions. Then we classified the synapse. And we taken the two criteria. The first one is the anatomical basis, and on that basis we divided the synapse into the three categories. First, we told that if the one neuron's axon rely on the another neuron's axon, okay, is termed as the axonic synapse, then axosomatic synapse, then exodendritic synapse. Okay, then we see in the functional classification, okay, there are two types of synapse. On the basis of functionality, there are two types of synapse. Okay, first one is very good. First one is your chemical synapse, second one is electrical synapse. Then we see, okay, the electrical synapse, they are rare in our body. Yeah, they are rare in our body, mostly present in the central nervous system. Apart from that, they are present in the cardiac muscles. Fine. They are also present in the epithelium of the retina, some of the smooth muscles, GIT smooth muscles. Okay, in these places there is the presence of electrical synapse. And the fundamental, you can say, the properties of the electrical synapse is that there is no any, you can say, the gap. There is no any gap between the one neurons and another successive neurons. Fine. They are connected by the gap junction. They are connected by a gap junction, and that we term as the synaptic delay. Okay, that we term as the synaptic, you can say, gap. Fine, the gap junctions. Then we seen another property of the, we can say, about the electrical synapse is that there is no any delay. Okay, the delay is very less. Fine, because there is a direct connection. Okay, there is a direct connection, so there is no any delay. Fine. Then we seen, okay, these synapse, okay, fine, the electrical synapse, it is rare in our body, so why we not encounter it? Then we seen, okay, if we refer the synapse, it means we are talking about the chemical synapse, because they are present widely in our system, in our body. Fine. About the chemical synapse, we seen that in the chemical synapse there is a gap that we term as a synaptic gap, or also called as the synaptic cleft, 15 to 20 nanometer in range. The space is 15 to 20 nanometers gap. Fine. Then we seen more things. Okay, then we seen already the more things. We seen, okay, the first neuron's membrane we term as the presynaptic membrane. Another, you can say, another successive neurons membrane we term as the postsynaptic membrane. Fine. Then we seen, okay, in the presynaptic membrane there is a vesicles, presence of the vesicle, that we term as the synaptic vesicles, and inside the synaptic vesicles there are chemicals, okay, very useful chemicals, we term them as a neurotransmitter. Fine. And then we seen for those neurotransmitter there is the presence of receptor on postsynaptic membrane. Fine, on the postsynaptic membrane, that we term as the receptor. And what happened actually, if we talked about the function, then these synapse, after receiving action potential, they will, you can say, the rupture. There is exocytosis of the neurotransmitter in the synaptic cleft. Fine. And those neurotransmitter which comes in the synaptic cleft, they will bind with their receptor and they form the receptor neurotransmitter complex. Okay, and that will generate the action potential in the postsynaptic membrane. Fine. But till now we didn't understood the mechanism. Yeah, we didn't understood the mechanism. So here today's lecture we will deal with, see the lecture outline. First we will deal with the function of the synapse, because we are left with the function of the synapse. Apart from the function of the synapse, we will see about that which category of neurotransmitters. Just remember, neurotransmitter is a kind of, you can say, the hormones, or it's a kind of chemicals. Okay, and it have both the activity. If it will, either it will inhibit the function, yeah, either it will inhibit the function or it will excite the function. So both the neurotransmitter are both inhibitory class and excitatory class. Okay, so we will see some of the names. Okay, we will see some of the names under the heading of neurotransmitter. Fine. Then the main thing is synaptic transmission. How, okay, the action potential which comes in the, you can say, the pre neurons, okay, presynaptic neurons, how they are transmitted, what are the process which we will see. And when we see these processes, no, we can correlate with our synaptic diseases. Fine, we will see the synaptic diseases. Then we see that there are factors which can affect this synaptic transmission. Okay, sometimes like the body have high acidity, called as acidosis. Okay, sometimes the alkaline nature increases, alkalosis. Okay, hypoxia. These can affect the synaptic transmission. So we will see also the factor affecting the synaptic transmission. Fine. And then without the properties nothing is complete. No, so we will also see the properties of chemical synapse. Fine, the properties of chemical synapse. Understood? So make the heading and start today's lecture. The function. Okay, function of synapse. As you understood, okay, as you understood from the previous lecture, that the main function of synapse is what? The main function is, first thing is provide one kind of junction. Okay, and what purpose, why this junction, what they serve for? They serve for transmission of, okay, they serve for, suppose this is one neuron, this is another neuron, okay, and they make one connection, connections. But why they are making the connections? We've seen that they make the connection for point-to-point coordination. Fine, for point-to-point coordinations. But what they are coordinating? They are coordinating the message, okay, which is, you can say, present in the presynaptic membrane. The white chalk, okay, take it close. This white chalk, as I am saying, it's a presynaptic neuron. Fine, and this is the postsynaptic membrane, and this is the gap. Okay, this gap represents the synaptic cleft. So whatever, you can say, information is present in these presynaptic neurons, they must be transferred to this postsynaptic membrane. That's the main purpose. So the main function is transmission of action potential. Write it down. The main function of synapse is transmission of action potential. Action potential is the message. Fine, no problem now. But this message can be inhibitory or it can be excitatory, isn't it? The message which is come, see, the central nervous system control our body. We are clear with this, no problem. Means they are controlling both the things, okay? Both, the, you can say, the, in a simple language, layman language, we can say they control the movement of our body, or they also stop the movement of our body. Fine. There is a secretion of the glands. Okay. Sometimes there is a secretion and there is no any secretion. Understood? So these are regulated. Okay. This functionality is regulated by the synapse. Okay. Or it can be inhibitory. Okay. It can be inhibitory. It can be excitatory. So it means we have to, if we talk about the function of the synapse, then we have to understand the two functions of the synapse. First one is excitatory function. It's excitatory function, and those, you can say, the synapse which is involved in the excitation, okay, excitatory function, we term as excitatory synapse. Fine. Understood? No problem. Now, the alternative is what? The alternative is inhibitory. Okay, so those synapses which involved in the inhibition of, you can say, one kind of function, they are termed as the inhibitory synapse. Fine, no problem. You can write it down if you want. Excited synapse means excitation function, and inhibitory synapse means inhibitory function. Okay, we will see that the inhibitory functions are classified. Okay, there can be direct inhibition, there can be indirect inhibition, there can be, you can say, the pre-inhibition, presynaptic inhibition, we will see. Okay, but for a while you just understand, if we talked about the function of the synapse, then it is not only about always, you can say, enhancing, okay, enhancing the function. It can be inhibitory as well, it can be inhibitory as well. So the function of synapse, the main function we have to keep in our mind is that they are involved in the transmission of action potential, or transmission of action potential, or we can say the message. But this message can be excitatory or it can be inhibitory. Fine, understood, no problem. Now once you understood the function, okay, once you understood the function, let's go with understanding some of the few names. We will have the detailed discussion over the synapse in our further lecture, but for a while we have to, in order to understand the, you can say, the synaptic transmission, how these synapses are, you can say, involved, we have to list down some of the neurotransmitter names, okay, which one category fall into the excitatory function and other category fall into the inhibitory function. So about the neurotransmitters, these are a special kind of chemicals, okay, these are a special kind of chemical which can either excite the neurons or inhibit the neurons. Remember, these are the special kind of neurons which can either excite the neurons or inhibit the neurons. Okay, so on this basis we are classifying the neurotransmitter into the two categories. The first neurotransmitter is excitatory neurotransmitter, NTS, short form neurotransmitters. Fine. Now inhibitory neurotransmitters. Okay, now see, inhibitory neurotransmitter, they are very less, few. Okay, so why not first we write down their name. The one name which you are familiar from your earlier classes is called GABA. Fine. Now GABA, and another is called as glycine. These two neurotransmitters, they are referred as inhibitory neurotransmitter because they are functioning. They are functioning. Okay, are you able to see it? The first one is called as the GABA, and another one is called as the glycine. GABA stands for gamma amino butyric acid. Gamma amino butyric acid. You should write it the full name. It's a gamma amino butyric acid. Fine, no problem. And if we talk about the neurons, okay, which are, you can say, the inhibitory neurons which possesses this GABA, they are present in the central nervous system, and we are referring those neurons as the GABAergic neuron. Okay, we are referring those neurons which are holding this GABA neurotransmitter, we are referring as the GABAergic neurons. Fine, GABAergic neurons, and they are present in the central nervous system. So if we talked about their location, they are present in the central nervous system. Now talk about the another class, glycine. Okay, it's a kind of peptide neurotransmitter. It's a peptide neurotransmitter, and if we talked about their location, they are present in the they are present in the spinal cord as well as the spinal cord, and very good, it is present in the brain stem and spinal cord. So write it down. It It is present in brain stem, okay, and spinal cord. So whenever synapse, okay, whenever the central nervous system, they are making the decision to inhibit the function, fine, in the central nervous system, these GABAergic neurons, they will fire, understood, and secrete the GABA if it is in central nervous system, or if it is in a brain stem and a spinal cord, they will secrete the glycine. GABA stands for gamma amino butyric acid, fine, no problem. Now let's talk about some of the excitatory neurotransmitter. The excitatory neurotransmitters, you can say the prime or most widely neurotransmitter which is present, okay, they are called as glutamate. Fine. Then we already seen one neurotransmitter. Can you recall it? Yeah, you already seen, no? Very good. It's the acetylcholine. Acetylcholine, or in short form we are writed it HCH. Acetylcholine. Then we also depicted in our previous lecture about the dopamine. Yeah, it's a dopamine, or we can pronounce as a dopamine. No problem. Fine. Yeah, apart from this dopamine, glycine, acetylcholine, there are other hormones as well, okay, and that is called as the your both neurotransmitter which are involved in the involuntary function, the autonomic functions. Are you recalling it? Yeah, it's your noradrenaline or adrenaline or norepinephrine or epinephrine, fine. So we can write it down, noradrenaline, understood, no problem. Now where they are forming, which place they are forming, okay, we know that they are stored in the synaptic cleft. If we talk about their storage, yeah, if we talk about their storage, both can be stored in their respective synaptic vesicles. Fine. If we talked about their storage, both are present in their respective synaptic vesicles. But where they are synthesized? They are synthesized, okay, by the help of transport. Okay, you will not understood until I will draw the diagram. So fine, I'm ready to draw it. Yeah, see, okay, fine. This is like fatty neuron. Okay, fine. So this is cell body, dendrites, okay, axon, and this is terminal end, okay, terminal end. The thing is, the terminal end, I already told you, they have the mitochondria, fine. Let's take the example of one, acetylcholine for a while, okay. The acetate molecule, acetylcholine is made up of acetate, yeah, plus choline. Choline is absorbed from the digestive system, okay, and they are transported to the terminal end of the neuron, fine. And acetate, it's a charged compound, charged chemical, and you know in the biochemical cycles, okay, acetate is synthesized. So acetate is synthesized itself in the axon, okay, and by the help of, yeah, mitochondria, fine. And choline is come, okay, and this acetate and choline combine and they form the acetylcholine. Are you understanding? So if we talked about their formation, they are formed inside the neurons itself. Understood? If we talked about their formation, they are formed inside the neuron itself. Now, once we understood about the neurotransmitter, let's recap once again. We are classified neurotransmitter on their functionality basis. Okay, if they are excitatory in nature, we list down some of the names. First one is your glutamate. Why I am written glutamate first? Okay, it can be asked in the MCQ. You just write it down. It is the most widely neurotransmitter, or we can say the most abundant. Put it the star mark over here and write it down. It's the most abundant. Fine. And if we talked about the acetylcholine, they are mainly present, you can say, on the neuromuscular junction. Okay, whenever the neurons make the synapse with the muscle, fine, there on that place there is the presence of acetylcholine. Fine, there is the presence of acetylcholine. And dopamine, it is present in the central nervous system. Okay, and I'm just giving you bit insight about this dopamine. What happened, sometimes these dopaminergic neurons, they become, you can say, the over functional and result into a kind of psychotic disease. Okay, result into a kind of psychotic disease. Okay, probably you heard about the disease. If we are talking about the synaptic disease, the disease which I am remembering right now is schizophrenia. Okay, it's a, you can say, the broad broad sign and symptoms holding disease. It's a broad sign and symptoms holding disease. Person have involuntary action, plus person have cognitive alteration. Okay, and they all, this kind of schizophrenic patient have all the kind of psychotic symptoms. Understood? And there can be abnormalities with acetylcholine. You previously seen about the autoimmune diseases that we term as the myasthenia gravis, isn't it? Myasthenia gravis. Apart from that, there are one more disease which is a rare, okay, which is a rare called as Lambert-Eaton myasthenic syndrome. Yeah, Lambert-Eaton myasthenic syndromes. These two diseases are associated with the acetylcholine, you can say, function, which we will see later on. But for a while you just understand. And if we talked about the characteristics, what happened to these myasthenic gravis patient and the Lambert-Eaton myasthenia syndrome, they have, you can say, without pain, okay, without pain, they are not able to, you can say, hold the normal body posture. Fine, their muscles, and especially those voluntary muscles, they become infunctional. First is voluntary muscles become non-functional, okay, but there is no pain. The characteristics feature about this disease is there is no any pain. Sometimes there will be a, you can say, sudden arrest. Okay, sudden arrest. It reminds me one more disease that is referred as a epilepsy. Okay, which we will see ahead. But for a while you just understand. And for understanding the neurotransmission help, okay, help the clinician to manage the symptoms of the disease. Fine, if there will be a problem in dopaminergic neurons, okay, if the problem is about the excitatory function, okay, then by the help of pharmacological agents, they can, you can say, inhibit the function, they can excite the function. Understand? Are you understood? Following? Yeah. So that there, you can say, why there are sometimes questions arises in our mind. Why we have to study so many things? We have to study the things because of their implementation. Yeah, because of their implementation. Fine. So that are the use why we are discussed this much, just because of their clinical importance. Fine. Now, once we understood about the neurotransmitters, yeah, we understood about the neurotransmitter. Let's go and discuss about the synaptic transmission. Okay, synaptic transmission. How the message is transferred from one neuron to the another neuron. And remember,